I'm really nervous, guys. What if the president doesn't like our ideas?
Don't worry, we should be fine as long as we cover everything in the points yesterday.
But I heard she was really hard to convince.
I did hear that from a colleague of mine, too.
True, but when you think about it, she has no reason to just turn us down. I mean, we do have a good idea, and she can make a lot of money off of it.
Right.
That's true.
Oh wait, let me connect this on. Is this the HDMI port?
I think it is.
Yeah,
That's right.
it looks,
Alright.
it's right there.
Cool.
So she did say she was meeting us at 1, right?
Yeah, I mean, it's 1:10 right now, so I'm sure that she'll be here soon.
2 hours later.
Yeah, I don't think she's showing up.
Me neither. It's getting pretty ridiculous.
Oh, look, she's coming. Get in position, guys.
Good afternoon, everyone. My name is Jennifer Ganwa. I'm sorry I kept you waiting for so long. I was running late. As you may know, I'm the president of Gilead Sciences. Formalities aside, shall we get started?
Good afternoon. I'm Sean Pacheco, and these are my colleagues.
Hi, I'm Pam England. I'm Todd Tran.
I'm Cody Boyer.
Together, we have conducted extensive research on the possibility of using triplex DNA technology in order to increase resistance to the HIV virus. HIV, as you may know, is a precursor to the fatal immunodeficiency disease, AIDS. So today, HIV infects over 1.2 million people in the United States. So how it works is, the T cells are important for activating other immune cells, such as B cells, memory helper T cells, cytotoxic T cells, and the HIV binds to the CCR protein, which is on the CD4 receptor on helper T cells. CCR5 is a G protein coupled receptor that allows HIV to enter the cell. HIV DNA is incorporated into the host DNA by using reverse transcriptase and integrase. It uses the host's own proteins against it. When enough of that virus is made, HIV leaves the cell and the cell eventually dies.
So as Sean was previously saying, the HIV virus binds to the co-receptor in order to affect the helper T cell. The co-receptor is called CCR5 and is actually controlled by a gene found on the human chromosome 3. Some individuals have a mutation called CCR5 delta 32, which is a result of a sequence of 32 base pairs being deleted. This deletion damages the receptor so much that the HIV can no longer bind to it. The mutation is caused by a recessive allele. This means if you have homozygous recessive for the gene, that you're going to have a mutated receptor so that HIV can no longer bond and you're completely immune to HIV. If you're heterozygous, this means that you have some protection, so if you are affected by HIV, you're going to have less severe symptoms. Unfortunately, though, if you still have a normal receptor, you'll be completely susceptible to HIV, which is caused by having the homozygous dominant trait. So now we're going to talk about the prevalence of the mutation. If you're Caucasian or of European descent, you're going to be 20% likely to be a carrier and only 1% likely to be completely immune to HIV. If you are of any other descent, you are not likely to have any of this immunity. The difference is due to natural selection, and Europeans were predisposed to bubonic plague and smallpox, causing this difference. The benefits are that you're going to be resistant to HIV if you do have this mutation, and if you have the mutation, you can also live a completely healthy, normal life.
So, as Pam said, people with the mutation benefit greatly from it because they are essentially immune to the HIV virus. So what we propose is a plan using the triple helix DNA to actually mute those 32 base pairs and then therefore emulate that mutation. So like as you see here on this figure is a triple helix DNA. Like what you have is the pre-existing DNA strands, which are anti-parallel to each other. So what we want to do is we want to use some technology to incorporate our own nucleotides in a parallel manner to one of the pre-existing strands. So like, even though it's going to be parallel to that one strand, it'll be overall anti-parallel to the farthermost strand. And yeah, in general, it would result in pretty much a mutation that is similar to the CCR5 delta 32 mutation, in where the person will then express proteins that are slightly smaller and then thus prevent HIV from entering and proliferating. So here is the gene sequence for the actual CCR5 gene, and as you can see in the red are the 32 base pairs, which are deleted in those people with the CCR5 delta 32. So if we get triple helix DNA, we can use nucleotides and then use them to complement those sequences, and then therefore essentially muting it from being expressed.
The actual process of our therapy, it's pretty straightforward. There are two ways to go about it. The first is you remove white blood cells from the patient, sequence their DNA to figure out the correct order of base pairs that you have to develop. Once you do that, you insert the DNA that we're creating to make that triple helix, and then that gene is going to be silenced. There's no more CCR5 protein expressed on the surface of the cell. HIV can't enter, the patient is immune. The second route is making our own cells, and we would follow the same process and then transfuse those back into the patient. In either case, you end up with a person who is HIV resistant. So now that we're done with our presentation, do you have any questions or concerns?
So, all in all, I do have to admit that your project has great potential, but I just can't fund your research since you didn't address these issues effectively. However, if you do find a way to work around these issues and solve these problems, I will gladly fund your research in the future. So, does that sound good?
Yes.
Sounds
Yeah,
good.
great.
Thank you.
Sounds fine, yes.
All right, I'll see you guys another time.
Thanks.
Thank you.
Thanks. So, we totally nailed it.
You know it.